I'd like to introduce Dr. Terschak. He got his undergraduate degree from the Rochester Institute of Technology and his PhD from the University of Alaska. We were just talking about it. He's one of the few people that gets to complain, or tells you not to complain, I guess, when it's 10 degrees below here in Chicago, because he has actually seen minus 65 degree weather, and that's without wind chill. So don't give him any grief about weather, right? Okay.
Yeah, my eyelashes, when I would blink, my eyelashes would freeze, and so it's this weird sticking sensation when you're blinking.
So without further ado, Dr. Terschak.
Thank you very much. I'm really, really glad to be here today. You guys are the first that gets to hear this talk about a newly formed undergraduate research laboratory called the Crab Lab. And that's what I want to talk about today, is this wonderful undergraduate experience that our IIT students are experiencing. And I'm also hoping that maybe some of you might get a chance to experience it as well. So if you're interested, talk to me afterwards, and we'll see what happens. I expect I'm going to have a nice long line afterwards. So the first question right out of the box is, what is the Crab Lab? And the Crab Lab is the creative research in animal behavior. Yeah, we actually chose the name and then fit the acronym first. So, and it's a behavioral laboratory, but here's the deal. I'm a chemist. I'm a chemist. And you ask, well, what's a chemist doing this type of biology? What you're going to find as your instructors teach you about your various different disciplines in science, you're going to find later on that every single one of them overlap with one another. And today's science projects cannot be done without the assistance of many different disciplines coming together in a collaborative effort. So those of you who think you're hardcore biologists and you don't care about physics, and so your grades might be slipping a little in physics, don't do that. All right, don't do that. You really need to have a strong background in all the different disciplines in order to be successful in your chosen field. So our Crab Lab is a research space for undergraduates. Not just undergraduates, but only undergraduates. We'll talk a little bit about that coming up soon. The purpose of the lab is to provide a space for the marine resources that the students use to do a variety of different types of research projects, all that take place in this one 25 by 11 foot room. So it's only, what, 275 square feet? Pretty small when you talk about the number of projects that are going on. You'll see about those in a moment as well. And then finally, it provides what I call a safe place for undergraduates to develop their own projects, their own line of questionings, to do research on something that truly interests just them. Not some professor who has a multi-million-dollar grant and is trying to pump out a lot of data so they can get tenure. No, this is all about the undergraduates asking questions that they have developed. So I want to start off by talking about this research space for undergraduates and undergraduate research. And my first question to you is undergraduate research important? How many of you think it's kind of important? How many of you think it's not all that important to get ahead? How many of you think it's super important? Yeah, a lot of you think it's super important. Well, some of you are right, actually. So the purpose of why it's advisable to have undergraduate research is because it's really a rehearsal for your career. You get a feel for what's really going on in your field and what it's like to do research, because taking a class and learning the material out of a textbook, or even going into a structured laboratory experiment, is very different than trying to do research, stuff that no one's done before. So it provides this short-term gauge for you to figure out your future directions. Whether or not graduate school is in your future, that's a major decision. I know a lot of colleagues who look back on it and say, boy, I wish I'd never done that. Those are the longest 7, 8, 9 years of my life. For those of you who know that you want to be a biologist, we know that there's other disciplines, subdisciplines within biology, or even chemistry, right? I'm an analytical chemist. But there's also organic chemistry and physical chemistry and inorganic chemistry and on and on and on. And so the research, short term anyways, allows you to sample from all these different subspecialties, these niches that your different disciplines have. But for those of you who aren't even sure if you're in the right field to begin with, this gives you an opportunity to say, you know, I really hate this field. I should switch to something that better suits me. And research gives you that opportunity. How many of you think it's a resume boost or an application boost, right? If you want to get a better job or you want to go ahead and get a application into graduate school for admission, how many of you think that research is really going to make it for you? Guess what? You're wrong. What? That was true 15 years ago. Today, however, according to a 2007 study by the National Science Foundation, that number's probably higher now. 72% of all the chemistry undergraduates, those undergraduates who are graduating, have had research experience. And so research experience no longer sets you apart. And this makes it even more difficult. Bucknell physics professor says, if you guys applying to graduate school don't have a letter of recommendation that specifically addresses undergraduate research experience, they assume that something was wrong. They assume that you're seriously handicapped. And so instead of your application going to the top of the pile, it now goes to the bottom of the pile. We'll talk a little bit about that in a moment, though, because I can see some of you already starting to panic. Yeah? I know it. I know it. Graduate school, it is important to stand out, but in order to stand out, you have to start really early. Okay? You have to start out in your freshman, even your sophomore year, and all of a sudden the freshmen and sophomores in the class are going, oh no. The reason for this is because the earlier you start, the longer you can commit to one project. And the longer you are in that project, the more responsibilities that you can take on. The more responsibilities that you can take on, the more likely your possibilities of generating data that will end up in a publication. Now, for those of you who are faculty in the audience, you've heard this publish or perish, and it's really about turning your results into papers. What I'm going to say is that's really not important for you as undergraduates to get on a publication. Sure enough, it helps, right? It does help. But the truth of the matter is what you're really going to get out of your undergraduate research experience is developing a relationship with your mentor. Your mentor is going to get to know your strengths. And for that reason, your research mentor is then going to be able to write an excellent, on-point letter of recommendation for you to get into graduate school. And a glowing letter of recommendation is worth more than a simple publication with 15 other names on it. Okay, it's personal. Also is the skills that you're learning as part of this research experience, because what they're looking for is that you already know how to conduct, I don't know, ELISA tests, or you know how to titrate, or you know how to use computers to generate or to process data and the like. So those skills are important. It's not just graduate school, it's also industry as well. Some of you are going to try to go right from your undergraduate degree right into the workforce. That's fabulous. Does undergraduate research then not apply to you? It does actually, and the reason why is because many of you, when you finish your degree and you go to the different online job postings, a lot of them are going to say that they want you to have on-the-job training or experience before you can apply for the job. The nice thing about this is if you've been committed to a long-term research project, a lot of employers will count that as job experience. And so you can waive that requirement. Right? Because it is. A lot of research is treated very much like a part-time or even a full-time job, depending upon how much time you can devote to it. So how many of you are worried? Yeah? That's okay. Because I'm going to talk to you now about how you can actually go about getting an opportunity. You guys here at DuPage. The first one is, I want to let you know that you're not limited to your own backyard. What that means is, I understand there's a handful of undergraduate research opportunities here at DuPage, but it's really hard to come by. Really, really tough. I understand that. So what you have to do is you have to get a little bit creative. You have to look outside your own little neighborhood. This is a variety of different places where you can get undergraduate research experience, and not specific, oh, this person's doing it, but you notice there's a lot of different opportunities out there that will count for you. often volunteer shadowing. This has to do with shadowing doctors. Those of you who are on the pre-med track, you know that shadowing a doctor during your summer vacation is a relatively important component of your application process to medical school. What I want to do is I want to talk a little bit about you having to go out and find a position, all right, finding research. And one of the first things you can do is you can go to a number of the different undergraduate and graduate schools in the Chicago area, or if you're willing to travel, you can look outside of your area, outside of Chicago, and go to these different universities' Office of Undergraduate Research, often abbreviated OURs, Office of Undergraduate Research, and see what type of appointments that they have. And then guess what you get to do? Get to write either a letter, stick a stamp on it, and mail it to whoever's doing that research and introduce yourself. Okay? Introduce yourself. That's the best way of getting your foot in the door. You have to create your own opportunities. Another one is summer REUs. You may have heard about summer undergraduate research experiences. And the National Science Foundation, the NSF, you just go to nsf.gov and you can search through and you can see that this has a whole bunch of different fields listed for our different REU programs that are out there all across the country. Because of that, you can search or limit it within certain states that you're interested in going to. All right, so that's out there. Keep in mind that summer stipend or summer research often comes with pay. It's not a lot, but it is nice. I think our IIT undergraduates get $5,000 for 10 weeks worth of work. That's quite good, isn't it? Yeah, I thought so too. But summer programs usually provide a stipend. However, if you were to continue the project during the academic year, that usually means that you have to take research credit and you don't get paid, but it does go on your transcript, most often as a science elective, or some undergraduate programs require that you have undergraduate experience in the laboratory as a credit before you can graduate. The last one is you got to go out and make your own. I made my own undergraduate research experience one summer because I had NASA Lewis Research Center in Cleveland, Ohio. Well, there was Plumbrook Station outside of where Cedar Point is. How many of you have been to Cedar Point? Yeah, right down the road. Instead of making a right onto US 250 to go to Cedar Point, if you make a left, there's Plumbrook Station, and it's their high hazard test facility. They make slush hydrogen. They had a nuclear reactor there. They test rocket engines. They've got the world's largest vacuum chamber. Hey, you guys waiting for the Avengers to come out, the movie? Yeah? When you go to that, SHIELD headquarters, when you watch the movie, SHIELD headquarters is actually the inside of Plumbrook's vacuum chamber, the world's largest vacuum chamber. There, you heard it here first. Okay? So that's what you'll see when you watch the movie now. Anyways, you need to make one. I went and I talked to the guard at the gate. He wouldn't let me on station, but he gave me the name of the human resources guy. I went home and I put together a letter and I drove back out and I handed it to the guard and I said, could you please see that he gets this? I got a phone call 2 days later asking for an interview. I made an opportunity happen. So can I say I'm a rocket scientist? No. I did environmental work out there, but that led to my choice of marine science and marine chemistry for graduate school because of this undergraduate research experience. See what I told you earlier actually matters. So how do you make one? You have to actually go knock on doors. Go meet with people. But before you knock on some professor's door, you have to make sure that you've researched their research. Go to the website, check out their website, look at some of the papers that they're publishing and get a feel for what they're doing. If it interests you, dig a little deeper. Because they're going to ask you 3 big questions. They're going to ask you 3 huge questions, and you had better have a darn good, well thought out answer in order to get yourself into their lab. The first question they're going to ask is, why do you even want to do research? Just general, not a particular project. Why are you interested in doing research? And you have to say something a little bit more substantial than, because I need it for my resume, or I need it to get into medical school. All right? Be creative and really find in your heart why it is that you want to commit to the hell that might be research. Okay? You have to commit to it. The next question that they're going to ask you is, why, so now that I know why you want to do research, why my laboratory? Why do you want to work for me? Yeah, you can do a little ego push in here, a little brown nosing if you want, but it really has to be thought out. You need to know what they're doing in order to say why you want to work for them. And then the last question they're going to ask you is, what can you bring to the laboratory? What skills do you have that allows that project to move forward? And if you haven't done research before, don't think that you're out of luck. Instead, what you can do is you can point to various courses and lab work that you've taken. Okay? You can talk about outside experience. Most times, what an instructor or a research mentor wants is not somebody who knows how to do all these procedures, but instead someone who's committed and willing to invest the time to not only learn the techniques, but also to stay with that laboratory long enough that the investment of teaching you how to do these things pays off for them in the end. Okay? A lot of times, the researchers would prefer to teach you how to do it their way rather than correct mistakes or habits that you've learned elsewhere. Everybody does things slightly differently, and so by training you from the fresh, they know that they've got you where they want you. You have to be prepared to commit to research. It is not some weekend project that you can fit in a half hour. All right? It's not like cramming for an exam. Research takes a committed, over time, consistent, dedicated effort. You need to be in the lab maybe 45 minutes a day, maybe a little bit longer. If I have time, I'll talk to you guys about how much time my research students are in the lab. Friday night, 9:30 p.m., I go downstairs to the laboratory. Did I say 9:30 at night on a Friday? Yeah, I was downstairs and all 3 of my undergraduate researchers were in the lab working. I told them, hey guys, it's a little late, you should probably head home. And you know what they said to me? Wait, one more trial, and then we have to feed. They were there until 10:30 at night on a Friday. Do they not have lives? Actually, quite the opposite. Quite the opposite. All right, so I want to talk a little bit here about the fact that it's only undergraduates. And I better pick up the pace here. I'm going to run out of time on you guys. So Jorge Sham, who is a cartoonist, he used to be a robotics professor at Stanford, and he found that he really enjoyed doing comics. And he has the lab food chain, and it is a delicate and balanced ego, not eco, but ego system. And he shows that there is God who is giving the money to the program manager who has all the money, to the faculty who sits in his throne, and before him, bows down before him is the postdoc. And then in the basement is the PhD student, cowering in the corner is the master's student, and then the lowly in the dirt worm of the laboratory is an undergraduate. Is that the way we really think of things? Well, you have to be very, very careful. You have to choose your laboratory wisely, and you have to look at the group dynamics as well. Here, this undergraduate has a mold sample in her hand and a mop. She's been cleaning up the lab that these guys made a mess of, and this guy is saying, well, I've done your taxes for you. Yeah? You got to be very careful. But this leads us to the next question, or the next thing to think about. It's a research experience versus a lab experience. There is a nuance. There is a difference between a lab experience and a research experience. If you don't go in knowing which one you're getting yourself into, you could be very disappointed. All right? Research means going in and having a project to yourself and trying to solve a problem and work around how to control for certain variables. It's lots of time, data crunching, et cetera, et cetera, et cetera. Lab experience means getting in the door, mopping up the floor, cleaning out the glassware, disposing of chemical waste, maybe getting rid of biological waste. In the crab lab, that means feces. Yeah. Is that something that you guys don't want to do? Well, I'm going to say this. Oftentimes, getting your foot in the door is really important, and you got to start at the bottom. And as you gain more experience and more familiarity with the different projects in the lab, you can take on larger and larger roles, bigger and bigger responsibilities. And before you know it, you've gone from washing glassware to actually being the one who creates the mess. All right, and that's what you're shooting for. Both of these are experience, and both of them are valuable. And I think everyone should start out with a lab experience once in their lives. Okay, makes you appreciate the people under you a little bit later. All right, so there are some difficulties with undergraduate projects. I'm going to cut this short. The reason is that undergraduates are fixed time, right? They're transient. By the time they're ready to do research, they're sophomores, maybe juniors. They come to me with a project idea and I say, how much money is it going to cost? And then we realize we don't have the money. So if I was to go the traditional route to apply for research funds, it might take 18 months. Well, what happens to a junior in 18 months? They graduate. I get the money, hooray! And the graduate's cap and gowned on his way out the door, and I don't have anybody to do the project. So what I need to do is I need to be very creative about where I can get my money. And this is one of the valuable lessons that my students have. They go out and they ask people for money. So what this means is they have to convince other people that have the purse strings that their project idea is important enough that they can give money to us to do that project. So what that means is the undergraduate has to be well-versed in the project, enthusiastic about the project, know it inside and out, and be excited about sharing it with somebody else. If you can get other people excited, they're more willing to give you money because they want to see the results to that question that you got them all excited about. There are some other difficulties with undergraduate projects. I think second to money is the time commitments. This is my large group schedule of just Monday and Tuesday. Wednesday and Thursday look the same. Friday morning is just as bad. And this is why the laboratory operates 24 hours a day, 7 days a week. Yeah, I've got stuff scheduled for undergraduates on Saturdays and Sundays. A psychology group comes in at 2 a.m. to set up a camera. 2 in the morning, folks, they're walking into the laboratory. So it does take a time commitment on your part. All right. So I want to talk about the facility itself, the Crab Lab. It started off as a small storage closet that once held radioactive waste. Yeah. We took this rather rough room, we cleaned it up. My undergraduates found $10 in paint and painted the walls on a weekend. Don't tell facilities. We did their job for them. We have a marine lab in Chicago. That's quite surprising when you think about how far away Chicago is from salt water. We have plenty of fresh water right in our back door, but it's almost 1,900 miles to San Francisco, close to 800 miles to Mobile, Alabama, and then Norfolk, Virginia is a whopping 700. All right, 700 miles away. So do we truck all of our salt water in for our species? No, we do what everyone does that has an aquarium. We go out and we purchase Instant Ocean. This is basically evaporated seawater that's been cleaned up, sterilized. But this one box here, 7.5 pounds, makes 100 liters of water. And you think, well, that's quite a bit. 100 liters of water, 25 gallons. But when you realize that our trials take 11 minutes, that's 10 minutes of actual data collection plus 1 minute of acclimation beforehand, running at 4 liters per minute, every single experimental trial takes 44 liters. That means we get 2 trials out of 1 box of salt. Every box of salt is $13, and we need to do close to 76 trials. You do the math. Yeah? We're going through something like a ton of salt and about $2,000 or $3,000 worth of salt. And it comes in and it goes down the drain. So we buy salt on a regular basis. $500 at a time gives us 36 boxes of salt. Lucky for us, we get free shipping. FedEx, not me. They say if you reach $45, they ship it for free. So I take advantage of it. Anyway, so you can see that we've got, that's 28 boxes sitting, or 29 because of this one here. That's 29 boxes sitting up in our shelf. That's all we can really keep in the lab at any one time. The rest is actually stored in my office. Here's my primary undergraduate. This is Kieran Shaughnessy. He's a 5th-year chemistry major. He's the one who helped and found the laboratory. He's making up salt. You can see we've got these 3 large 55-gallon barrels, drums. Basically, they're trash barrels that we bought from Home Depot. He's pouring in salt. You may notice that we have a hose that goes from regular tap water. If you've ever worked with fish, they tell you have to let the water sit because of the chlorine in it, right? Well, not only the chlorine, but there's also a lot of metals dissolved up in the water, including copper. And copper will harm our octopus. Yeah, I said it. I said an octopus. So what we do is we have to treat the water ahead of time. It's a chelating agent. Those of you who've had chemistry, you may know a little bit about chelating agents. Basically what it does, it instantly ages the water. We have to add about 60 milliliters or 2 ounces of this to every 100 liters of water. Our seawater system, we actually have 2 closed-loop systems. The first one takes care of the European shore crab, also known as the green crab, Carcinus manus. And here's one of our lovely guys. You can tell how bright green he is. They are rather ferocious. If you get your finger caught in one of those, it will hurt, believe me. Here's the front end of them. This is communal housing. And so we have 60 of these guys, 60 of them. 12 of them are test species for the trials. The others are food, not for us. They don't have any real value. In fact, they are a globally invasive species. How many of you guys know what an invasive species is? Yeah? Okay, we're going to talk about that a little bit when we look at one of the hypotheses. So this is our seawater system. In order to make sure that we have 400 liters of seawater, 450 now, 450 liters of seawater, we have these ballast tanks. They're all connected by through-hull fittings. They go to a regular aquarium pump with a filtration system in it. We've got chemical filters, we have particulate filters, we have ammonia filters in those. This basically keeps the seawater quality where we need it in order to sustain the life of the crabs that are living in these bins up here. The bins on the left, these 4 here, are communal housing. That's where we put the food. These 2 bins down here have subdividers in them, and that allows us to keep our different test subjects separated from one another, because you can't name them. Well, we've named them, but you can't put a name tag on them. You can't color them because they rub it off. And so if they were all living together, we wouldn't know which crab was part of which set of trials. We also have 2 10th horsepower chillers. As things start warming up, we have to make sure the crabs are kept at a nice, cozy, cold temperature of about 11 degrees Celsius. All right, and so that's about 54 degrees Fahrenheit, and that's the temperature that they thrive at and they do very, very well at. This is a picture of the individual housing for these guys. You may notice the containers are Home Depot storage bins, and the dividers here are diffusers for fluorescent lights. Yeah, so next time you look up at fluorescent lights and you see that grating, we actually broke it up and turned it into housing dividers. We also have a giant Pacific octopus. This is the same type of octopus that the Shedd Aquarium has. If you've ever been to Shedd Aquarium and you see their octopus, it's Oprah. Their octopus is about the same size as ours, but they've had theirs for a little bit longer than us. Isn't he beautiful? He's my baby. I love him. And to give you an idea, he's about 1.1 kilograms in mass. His head, if you take your fists and put them together, that's about the size of his head. And if you stretch one arm, not a tentacle, arms, one arm to the other arm, he has a maximum span of 32 inches. Yeah, pretty big. He is so fun. He is so social. In fact, Shedd Aquarium's a little jealous because our octopus comes out to visit with people, enjoys having people around, so he doesn't hide very often. This is his setup. It's the same type of filtration system here. We have a larger pump system, or we have a larger chiller. It's a 1/2 horsepower. And this keeps his temperature at 11 degrees as well. The reason why we have a much larger chiller on it is because if you start reaching temperatures of 13 or 14 degrees C, then the octopus starts having health issues. They don't like these warm temperatures. This is a 120-gallon aquarium tank. It's 48 inches by 24 and 24 tall. We have some flower pots here to serve as dens. He has one of his favorite toys in there. You may know that octopuses are quite intelligent in terms of having need for enrichment. And there he is. Ha, I love him. Anyways. We also have a series of sensors that monitor water quality all the time, and this bundle of wires with all these boxes over here takes all the dissolved oxygen data, the pH of the water, the temperature of the water, and the conductivity or the salinity, how salty the water is, and it feeds that to the internet. And so I'm able to monitor in real time what the conditions of the laboratory are anywhere in the world. And guess what? You guys can do it too. And I'll show you how to do that at the end of today's talk. This also has an alert system. If any of those different characteristics go outside a prescribed range that I've set, it'll not only send me an email, it'll also send me an alert text message. And it sends it to everybody in my group as well. And we all converge on the lab and try to find out what's wrong, even if it is 3:30 in the morning. 2 weeks ago, I was in the lab at 3:30 in the morning because we had some heat issues. Anyways. That system also puts the laboratory on a 12-hour light and a 12-hour dark cycle. We are in the basement, there are no windows, and therefore the crabs, which use light to regulate their rhythms very much like we do, we have to make sure that their light and dark periods are available to them. We have daytime start at 2 a.m. in Octoland, as we like to call it. And the reason why is because my undergraduates' research experiments have to take place at night, and rather have them here at 3 in the morning, I've actually got them in the laboratory at 4 in the afternoon, which is dark in the laboratory. So the crab and the octopus all think it's midnight when it's really only 4 in the afternoon for us. So, so what we have is we have these full spectrum LEDs. So this is daytime with all the white lights going, and at nighttime, if you can see that, it shuts down all the white ones and turns on some blue ones, and that simulates moonlight. And everybody's happy, except for psychology, who's trying to film in the dark. All right, so the last thing that I want to talk about is this safe place for undergraduate research. And what I mean by safe is my students, they design their own set of questions. I don't give them a project. And what this does is it really instills a sense of ownership. These undergraduates own their projects. And the reason why that is so important is because if you have a vested interest, if you're really excited about what you're doing, you're going to be more committed and more dedicated to seeing the outcome of that project. I've told you before that my students don't want to go home. It's because it's their project. They're generating data to ask or answer their questions, not something I posed to them. And so we've got commitment and dedication coming from ownership. And what I mean by a safe research environment is just that. My students have the freedom to struggle. They have the freedom to try things and make mistakes. Part of the reason why this is not always the case in other research labs is because whoever gets the money is beholden to the granting agency. Whoever gave them money is demanding results. And so that professor is under the gun all the time to make those dollars turn into data to satisfy the granting agency so that way they can go back for more money later. Not only is it about generating data and producing results in that laboratory, a lot of careers, that is your faculty members, your tenured professors, in order to get tenure, they have to show publication results. And so their livelihood really depends upon data being generated in the lab. If you're working for somebody who's under the gun, how much leeway are they going to give you to make mistakes? Not much, especially when every dollar counts. And they want to make sure that they give you projects that you can be successful at. That's the nice thing, right? They're going to give you projects that they know you can do. So just be aware of that. What this means, though, for my group, because they have the ability to fail, I have to make sure that the projects that they're choosing are not only interesting, because we want to make sure that we're generating data that people care about, but we also have to make sure that those projects are designed in such a way that they're actually able to be successful. Are they doable? Now, the reason why that's over here, and we've got to balance that, is because there's some other equation in the mix here is that's also that we have to balance those needs against the resources that we have available to us. It's a marine laboratory. All right? We really can't be doing much with freshwater fish because everything we have in the laboratory is saltwater. And so that would mean that we would have to design a whole nother system, and that would cost more money, and et cetera, et cetera. So we have to try to keep the projects within our capabilities of the resources available. And that's sort of what my job is as the laboratory facilitator. I like calling myself a facilitator because my job is to say yes to the students as often as I can. I'm not looking to tell you guys no. I want to say yes. So what I do is I help the students design projects that get at their questions such that they meet the resources available in the laboratory. All right, so let's talk about the research project in the last 10 minutes or so that I have left. Is that right, Tom? About 10 minutes? Yep. Okay, excellent. So we have 5 different groups going on. We've got a chemical ecology group, which is my primary group. We have a cognitive science group. We have endocrinology going on. We even have some renewable energy, believe it or not, and microbiology, which is kind of in the works. For those of you who aren't sure what these different groups are, chemical ecology is basically how both plants and animals respond to chemicals in the environment. Most people think of chemical ecology as being pheromones. All right, you guys all know what pheromones are? Yeah, sexy smell? Yeah, that's exactly what it is. It's signals, they're odors and chemicals that are released into the environment that living creatures, and I also count plants because plants can know where to settle and know where to grow based on chemical signals coming to them from the environment. My job is to try to find out what those chemical compounds are and also put an environmental slant on them. For example, one of our projects looks at a pheromone that the crabs use, and we're trying to track how global climate change is going to acidify the oceans. And with dropping pHs, does that change the structure of the pheromone so much that the crabs no longer recognize it? Yeah, good question, huh? Yeah, I like that one too. We're not talking about that one today. We've got something more interesting than that. Cognitive science is the psychology department's group, or Institute of Psychology. They're looking at how octopuses play and whether or not they play. Play is a strict definition. It has no other purpose, recognizable purpose. And also, because these guys are fairly intelligent, it's problem-solving skills. So if you go on the website and you start watching some of the video feeds, you're going to see the octopus playing with toys. You're also going to see him solving puzzles. Yeah, just YouTube, Google octopus problem-solving, and you'll see all sorts of neat puzzles that octopuses are very clever about getting at. Endocrinology, this is the study of hormones and such in the body system. And what we have is we have a project going on which is looking at stress indicators based on other things that were going on in the lab. Remember I said that we had a temperature problem the other week? Well, we've taken fecal samples from the octopus and we're going to be studying the hormones that have been excreted in that feces in order to determine whether or not how much stress he was under. More importantly, though, we're also tying this to the cognitive sciences problem-solving skills. When you present someone with a puzzle for the first time, it's kind of frustrating, isn't it? But as you play with it and you get to know how to solve it, you solve it in less and less time, and it's not so stressful. We're hoping to see the stress hormones drop off as the octopus becomes more adept at solving that particular puzzle. We are also growing algae biomass in the laboratory. It's a salt water. They basically use our wastewater, so nothing is just immediately thrown down the drain. So they're going to be growing different types of algae and seeing if it can be turned into renewable fuels, renewable energy. It's part of an iPro. We'll talk about that in a moment. And microbiology, the project is still in development. I'm not a microbiologist, so if there's any of you who do microbiology and you've got a neat idea, love to hear about it, okay? But we're looking at populations, densities, dynamics, nutrients, all of that. So in chemical ecology, I've got chemists and 2 biologists. CogSci has 3 psychology and 1 mechanical engineer because they need somebody to design the puzzle boxes. Molecular biochemistry, 5th year just joined us. He's awesome. Faraz is great. He's really super dedicated to the laboratory, as are all the students, actually. We have 4 students with iPro 328. This is this engineering primarily project that is looking at biomass from algae, or biofuels from algae. And we have 3 microbiology students who are trying to put together an undergraduate project in that field. So that's 15 students, 5 different projects, and more of them keep coming in every day. So as you might guess, it's hard to divvy up the cash. It's also hard to divvy up the resources, but we're making it happen. All right, I want to talk about the chemical ecology one very, very quickly because I know I'm running out of time. The Carcinus manus, the green crab, is not native to the United States, and that's what our colors here are. You can see where it's originally native to, and that's why it's often called the European shore crab. But through shipping, in the 1800s even, sailing ships brought these crabs over to the east coast of the United States, and they've taken root. They decimated, they ate all of the clams. And so clams are a little bit harder to find now on the eastern seaboard. Notice that with the opening of the Panama Canal, eventually we started seeing green crabs on the west coast. In fact, in 1990 was the first instance of green crab appearing in San Francisco Bay. Since 1990, what's that, 20 years, they've already spread all the way up to British Columbia, Vancouver area, and they're continuing their march out the Aleutian chains. They disrupt the native ecosystem because they seek out food and they also take shelter away from the native species. So that added pressure is making it hard for the native species to survive. They're also a threat to the mariculture and the fishing industries. Because they're taking up resources, juvenile crabs, like the Dungeness crab, right? So the commercially important ones on the west coast are having a more difficult time becoming adults. And that also means that the fishermen are having less catch. And so we want to make sure that we're finding ways of controlling these guys. Mariculture is sort of oyster farming and mussel farming in bags and such. When those crabs get in there, they can eat all of it. In fact, there's one guy in Tamales Bay who lost $4 million worth of product one year due to the invasive green crab. So all of our measures to control these guys have failed. We tried fishing for them. That was ineffective because it was too many man-hours to do it. They've tried to use pesticides, but they were non-discriminatory. That is, they killed lots of other things as well, plus it wasn't good for human health. And then they tried biological methods, like using parasites to feminize the males. It turns out that that wasn't species-specific. So they started feminizing all the crabs, and that's not good either. So what I wanted to do is I wanted to take an integrated pest management approach. I wanted to use natural products, that is chemicals already in the ecosystem that controls behavior. For example, reproductive behavior. The boll weevil, right? This is a successful employment of pheromones to draw boll weevils out of cotton areas and corn. Those of you who are familiar with that agriculture industry know that this is a very effective technique that is easy on the environment. I wanted to try to apply that to the marine environment. It's only been successfully done once, and it was in the Great Lakes with lamprey eels. What we're doing is we're taking our green crab, and we have to ask ourselves, so what will draw a green crab out of an area? Well, food is one thing, but that also draws everything else. What about a female crab? If he finds a good-looking mate, then he'll be drawn out of that region. And that was my postdoc research. My postdoc research, I was part of the team that found this molecule. Sorry, it's a little hard to see under these lights, but this is UDP. How many of you guys know what UDP is? Uridine diphosphate, right? A nucleoside. Cool, huh? Small molecule. This is fabulous. And so what we did was we found that these male crabs are interested in following this scent odor, this trail, to get out of one area into another. If we put that in a trap, or we put it someplace where there's not a lot of females, it means the males won't be successful in reproducing, and therefore the numbers start dropping. But if we've got something that draws them, what happens if we find something that pushes them out of an area too? Isn't that a doubly effective method, right? Push and pull? Yeah. And so what we thought is, let's find something that eats them, and it happens to be an octopus. And so our hypothesis is that there is some sort of chemical compound out there that the octopus has associated with them, an odor that the crabs can smell and say, oh my, I smell an octopus, I'm getting out of here. And then they leave. The sub-hypothesis is that this chemical that they eat is part of their metabolism. It's not something they excrete, but it's something that they digest and excrete from their waste products. So we're going to feed this octopus a diet of nothing but green crab. The idea here is that if the green crab have an odor after they've been eaten coming out of the octopus, then the green crab are really, they're like, oh man, I smell that he ate one of my friends. I know he's hungry for crab. I'm a crab. I'm nowhere near here. So that's the first sub-hypothesis here. And then what we're going to do is we're going to switch his diet later on to nothing but mussels, and we're going to see if the crabs respond differently, see if they are a little bit more receptive to crab-fed octopus as opposed to mussel-fed octopus. So what we do is we have a test flume, and a bunch of water flows over this, and we've got it measured. It's 300 centimeters, so about 3 meters long, 10 feet-ish. And at one end, we have a series of pumps. And these pumps pump water from the octopus and from the crab at different times. We flip a switch. And that water flows in at one end, and it's a laminar flow, and we've put a crab in the middle. But in order to make sure that he starts out in the middle, we put a little cage around him. And once that cage is in place, we start the pump going, and this is pumping crab water, the water that he's lived in for the long time. So he's happy about living in that water. He's not sensing anything dangerous. He's going about his business. And then we remove that cage, and we see how he kind of walks around when we switch from the crab water to the water that the octopus has lived in. And what we measure as this crab is moving around due to this new water that's flowing in, we're going to go ahead and we're going to look at how quickly he moves in that flume, both upstream towards the smell of the octopus, as well as downstream. What do you guys think? I'm an octopus and I smell a predator. What happens? How fast am I going forward? Upstream to where the octopus is. Am I going really, really fast because I want to go and meet him? No, I'm going really slowly. How about if I smell that octopus, what's my nature? What am I want to do? Right? I want to run. All right, so already you guys have this hypothesis that they're going to move slowly and cautiously upstream versus really fast downstream. That's if this chemical compound is present, because there's no octopus up here, is there? They can't see the octopus, so it's not visual. All right. The other thing that we look at is how much time they spend resting upstream versus downstream. You guys know the fight or flight response, right? Well, there's also a hide response. All right? And crabs are really good at hiding. So what we do is we designate the front end, 30 centimeters, as being upstream, and the last 30, which is downstream. And we say if the crab stops and hunkers down and doesn't move, we start a timer and we calculate how much time, or we measure how much time he's spent resting in the upstream versus the downstream. If I'm an octopus, or sorry, if I'm a crab and I smell an octopus, where am I going to hide? Am I going to hide really, really close to him, or am I going to hide really, really far away from him? Far away. Okay, so there we are. So let's take a look at these results. These are all preliminary. We've only run 24 trials, and you might say, well, that's not very many. When you talk about every other day, we can only run 3 to 4 of these. I think 24 is pretty good. The octopus has been in the lab for 6 weeks. We've only had him for a month and a half. So here's the first set of results. Our foraging velocities, this is the speed upstream versus downstream. So his speed upstream, this is the orange bar is the octopus conditioned water versus the control water. And if you notice, there's about 100 centimeters per minute difference in their advancing velocity. So sure enough, just like you guys predicted, they actually travel upstream at a slower rate when the octopus water is being flowed over them versus upstream at a higher rate when it's just the water that they're used to living in with all their friends. Does that support our hypothesis? Yeah, it sure does. How about in the opposite direction? That is when they're going downstream. When they're going downstream, notice they're moving at a much faster rate. Again, 100 centimeters difference between them moving downstream away from the octopus when they smell him versus his regular speed downstream, which is, you know, a little bit slower. You might ask, well, why is there a difference between upstream and downstream? That's 100 centimeters difference as well in the control water, right? Their natural instinct is to travel upstream. The reason why is because they can smell ahead of them. Right? For those of you guys who are hunters, right? You always want to stay downwind of your quarry because you don't want to spook them. Yeah? And so the crabs are they're always sensing, and if they don't smell anything bad, or if they smell something good, they're always traveling upstream because they don't know what's behind them because the scents are being carried away. Yeah? So they're blind in one direction, if you want to call it that. Average time spent resting, I was going to cut to the chase. Notice that when it's upstream conditioned water, or octopus conditioned water, they spend a lot less time than they do if it's in control. That's what we said, right? They don't spend a lot of time hanging out where all this octopus water is coming from. As we might guess, they spend more time resting downstream than they do upstream when that octopus water is flowing, compared to controls. Yeah? Does all of this support our hypotheses? Yeah, it sure does. I'm excited by that. All right, so we have a strong indication that there is something in the octopus conditioned water. What we're going to do now is we're going to continue in the laboratory with testing crab-fed octopus water, primarily for statistical purposes. You notice I didn't put any error bars on that, just because we don't have a lot yet. But what we do have doesn't overlap, so we're feeling pretty good about it. Then what we're going to do is we're going to take the octopus water and we're going to just boil it, not the octopus. We're going to remove the water first. We're not making taku, right? Sushi lovers? Yeah. Anyways, the day after we got the octopus, I went out for sushi and I had some. I thought that was kind of ironic. That is ironic, isn't it? That is ironic. So we're going to denature that protein. If it's a protein, we should see that the water no longer has activity for it. And we can say, aha, it's a protein because we denatured it. And then we can focus our search on proteins. Barring that, what we're going to do is we're going to use solid phase extraction. It's sort of like a chromatography method. It's a chromatographic method, but not really. It's more extraction. For those of you who have done organic chem, you may know about SPE, solid phase extraction. There's more information on the website about it. But basically what we're able to do is we're able to break the chemicals. We're able to separate the chemicals from each other based on very broad categories. This hydrophobic acids, both very and slightly, hydrophilically charged acids, as well as the hydrophilically neutral compounds. And we're going to test each of those just to try to narrow down what type of compound it might be. And then from that, we can use HILIC, which is hydrophobic interaction liquid chromatography, in order to identify that compound so we know what it is. Of course, we're going to change the diet to mussel-fed when this is all done. You may know them as the blue mussels. This is the same ones you get in the grocery store, in restaurants. And then finally what we're going to do is we're going to change to an Atlantic species. We're going to change out our octopus, no longer using the giant Pacific octopus, but instead we're going to look to see if the same sort of results come up with the common octopus. And that's this guy here who's found in the Atlantic Ocean, all around Florida, etc. All right. Final thoughts, and then we'll turn it open for questions. The Crab Lab is a student-initiated endeavor. The students come to me with ideas, they built the laboratory, and they keep it running. But I want to point out that there's more to science than simply going into the laboratory and taking data. There's a lot of fundraising that has to take place, and my students learn this the hard way because they have to go out and find funds. Fortunately, it's made easier because they're using CMFs, charismatic megafauna. Yeah. Everybody wants to become a marine biologist because they've seen dolphin movies or Shark Week on television and think those are really cool. Got news for you guys. That's not what marine biology is anymore. All right, we're not dealing with cute cuddlies or ferocious beasts. What we're doing is we're doing molecular biology, genetics. Gone are the days when you're doing taxonomy or dissection. That doesn't work anymore. You're no longer beach combing. That's more ecology. But the other part is that you have to instill interest. You have to inspire other people to want to know the answers to the questions that you're posing. What that does is not only does it keep some public going, but it also brings in the money. Speaking of money, you have to communicate how your progress is going. We do it in a number of different ways. This is the Tech News. This is the IIT newspaper. We've got 2 articles out already. We're just released today in IIT Online. IIT Magazine is going to be doing a feature on us. I even heard that we may be doing an NPR spot. Clever Apes. You guys familiar with Clever Apes? Yeah, there's a possibility. Our marketing people are trying to get me and Kieran, my undergraduate researcher, on to Clever Apes. I'm really excited about that. Anyways, you can follow all of us because part of our communication is also to the general public. We have a website, is.gd. I always remember is good, Crab Lab. Go online to that, and what you'll find is right now on our homepage is an octopus naming contest. We don't have a name for the octopus. But right now, Ollie is leading. So if you're interested, go online, vote for your best. Hopefully, my undergraduates maintain the website. Hopefully, they fixed the glitch where now you can go and see the pie chart that tells you who's ahead and who's behind. Another thing that you can do is follow us at a Twitter account. We have a really interesting thing because we've got 3 separate Twitter accounts from the different points of view. The crabs tweet about whatever they're doing. The octopus tweets about what it's doing. I think one of them here, this indicates that last week we had some temperature problems in the laboratory, right? Cold, warm, hot, 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 cold again, thank you, all right? Because we instilled a new chiller. So yeah, you can follow along what's been going on. And notice here we are, Dr. Terschak presenting at Ecology DuPage about our octopus. There you guys made our Twitter feed. Woohoo! All right, go you guys. This is also where you can take a look at all of the stats online and follow what's been going on for the past day or so. And you can always keep an eye on my students as they work. So that is what I have for you, and I would love to hear your questions in a short amount of time. I have no place else to be after this. Well, I do, but I can stick around and answer questions if any of you guys want to stick around afterwards. Okay? So I would love to have questions.
Thank our speaker.
Thank you.
And then we'll open it up for some questions.
So the question is whether or not we know what would happen if we introduced a whole bunch of octopuses into an area. Would it drive crab out? We're not looking at that per se. What we're hoping to do is once we identify the chemical compound is to make it synthetically so we don't actually introduce a whole bunch of octopus. They're fairly territorial. The only time octopuses get together with one another is when they're ready to mate, and when they mate, it's really close to time for them to die. They mate at the end of their lifetimes. So yeah, actually trying to put octopus in one area would not be good for the octopus, and it really wouldn't be good for the other food that they eat. And so that's why we're trying to look for the chemical compound. Yes, we're just going to introduce the chemicals. Yep. So the question is, are we going to test any other species that might be natural to the, it is eventually going to be part of the project. We need to identify that chemical compound first because it has not just a chemical ecology importance, but it's also important to the evolutionary biologists to know what those chemical compounds are. Other ecology species, for example, the UDP that we found, which is the pheromone for green crab, also work on a number of other species and a number of other genus as well. And so it's a real question about where did these, was it an evolutionary diversion, or is it something a little bit more basic to their biorhythms, their own personal biochemistry. So yes, there will be a test, but that's much further on down the road when we're looking to deploy it, because we definitely need to see how it affects the rest of the ecosystem as well. We don't want to cause more damage than we're fixing, so yes. So the question is whether or not these octopus play. Can you talk about the toys? You know, there's some question whether or not what we're seeing with him is actually play or not, because play has a very strict definition. It has to have no other function. For example, when you see baby cubs, like tiger cubs and lion cubs wrestling with one another, or young stags butting heads, well, that's really not play. That's more figuring out life roles and practice for other things. You watch your cat play with a toy all the time, but what's he really doing? Stalking, right? But what we're trying to see is whether or not there are play in the octopus, because they are fairly intelligent. When we say intelligent, it's because they have a larger nerve mass to body ratio, and therefore it kind of tends to indicate that they do have some higher level of intelligence. We're not talking being able to do math, although I'm sure it's base 8. Sorry. But we've seen him actually investigate some of these toys. Whether or not he's looking for food or actually playing, that's the cog size group, and they're still processing data. I'm hearing good things from them. There's anecdotal evidence that octopus at different marine aquarium would use, because they use a jet, right? They can shoot water, they can use for propulsion. Well, one of the octopus would have a floating ball, and they would squirt water at that ball, and it would get caught in the current, and basically what they would do is they would keep squirting that ball and playing with it. It didn't serve any other purpose. And so we're just trying to look at these others. So he has some toys that he's familiar with. He has this football. You may have seen that football in the tank. It's this rubberized plastic mesh football, and the idea is just stuff a piece of food in it. Is he playing with the ball, or is he going after the food? Probably going after the food. But if there's no food in it, is he searching for it? And what else is he doing? Lots of different questions. We've seen him without anything, just run his arms through all those holes, whether he's searching for something because they taste with their suckers, or whether or not he's just trying to enrich his environment. It's a question. He certainly recognizes people, and he has this personality. When Kieran walks into the room, he can hear Kieran's voice, and he'll come out to meet Kieran. It's really weird. When I go down there, sometimes. When Miriam and Mariah go down, he'll come out for them. Often when some of the cog sci group come out, he kind of stays really still. and then eventually comes out of his shell, or not shell, but out of his dens and such, and he starts interacting with them. So some people he warms up to immediately, and others he takes a little bit of time. Interesting enough, though, we had a big photo shoot in the laboratory for some marketing stuff, and we had like 6 or 7 people in the laboratory. There was a lot of commotion going on, and he was loving it. He was a perfect color. He was spread out on the blue gravel, and he was all textured up, which meant he was happy, and he was just doing all sorts of great stuff. Hamming it up, actually, is what he was doing. So they can change. Not only can octopuses change. So the question is well, what does texturing up mean? Well, not only can they change their color, right, because they have 3 basically sacs, ink sacs, not a jet ink, but a chromatophore type ink sac, and they can close and open these to get different color and patterns on the surface of their skin. It makes them look like rocks. It makes them look like their bottom. So he can go from a really pale white color to a very deep cherry red color and everything in between. Most often he's this brownish red color. That's his normal color. But here, their skin, they can also change the texture of their skin, whether or not it's this very smooth and glassy looking surface, or if it's got all these little bumps and horns on it. If you notice, he's got this mottled color, and if you notice above his eyes, you see those little pokey things? We call them horns, but he's able to make those go away. And so this rough texture is actually a sign that he's healthy, happy, actively engaged. And that's what we look for. We look for this textured skin, and we look for a brownish red color. If he turns white, it may be that he's trying to camouflage himself with some of his background, but it also is an indication that he's unhappy. When I say unhappy, we mean either too hot, not feeling well because of other water quality issues, and we try to really figure out what's going on with him. And most of the time we find out that that's just the color he wants to be. For example, if you go to Shed Aquarium's octopus and you take a look at Oprah, she very often is this pale white color. It doesn't mean she's unhappy or sick, it's just the color that she chooses to be because of her surroundings. If they turn this bright cherry red, it usually means they're angry. We've seen him once like that, and that's when he arrived, because he was shipped from British Columbia, from Vancouver. He came in a Coleman cooler. We opened it up, and he's in an oxygenated environment, and he just was a little scared, as you might be, right? Somebody just opened the lid on you. Even if the lab was darkened, it was still a bit of a shock to him. And we got him in the water, and as soon as he got in the water, changed back to this brownish red color, which indicated he was happy. So. The question is whether or not octopuses can get sick, and yeah, they can, actually. They have their own disease. They have their own disease-fighting mechanism. So what we have is we have a lot of protocols in place to make sure that we're not introducing any sort of germs, bacteria, foreign objects into the lab. A lot of it has to do with sterilization. Everybody washes their hands up to their mid-arm before they put their hands in into the tank, both before and after going in. All of our tools and implements are sprayed down with just a weak solution of Clorox bleach, but then they have to be thoroughly rinsed before they go into the tank, because the bleach can hurt them as well. So yeah, everything is really monitored in terms of making sure he doesn't get sick. Excellent question. He asked if there was any, if we've had any trouble with him trying to escape. If you take a look at his tank here, it's really hard to see here, but what I've done is I've built a octopus-proof lid on it. So what we have is we have these 24 by 24 inch panels that slide in through sort of like tongue and groove, and then the front is tongue and grooved, and the front panel gets on, and then we take clevis pins, and the whole thing is actually Gorilla Glued to the top. So he would not be able to get out. However, we also, I don't know if you noticed this sort of frosted area here. That is carpet runner, that clear carpet runner, that's smooth on the top, the plastic clear stuff that runs along a hallway. On the back side, they've got those little pokey things in order to keep it from slipping. We basically took it and we mounted it such that the pokey ends were out. And so if he tries to climb up into this area, it's uncomfortable. He doesn't like it. You'll see a lot of aquaria try to use astroturf, fake grass, and that's because they don't like the texture. problem is a lot of these astroturfs have antimicrobial agents added to them, and that will make the octopus sick. And it also collects a lot of debris, and it allows algae to grow, and we just decided in order to avoid all that, we were going to try this. So not only do we have the first 4.5 inches around the top of his tank covered with this, what I call anti-octopus barrier, we also wrapped the outflow tube. So we have a siphon tube. We've wrapped that with the anti-octopus barrier, and we also covered this back wall in this area where the sensors are. This ball here that you see, this black ball, that's actually the dissolved oxygen sensor. And if we move it around, it's about the same size as a crab. So what does he do? He goes after it. He loves playing with this black ball because he thinks it's food, and eventually he lets go of it. And so in order to keep him from doing that all the time, we've kind of put the back wall with this stuff as well. The question is, does he grab onto people's arms? We try very hard not to touch the octopus. It won't hurt him, our touching him, but we just really are trying to avoid a lot of excess contact with him, more for his health than anything else. But there's a couple of times where we've just checked to see if his suckers were doing all right, and so we put a wet hand in there, and sure enough, yeah, he would grab onto a finger, and then he would start wrapping his arm around, and we'd pull away, and he's pretty strong for his size. He actually is.
Let's thank our speaker one more time.
Thank you very much. Excellent questions, too. So, all right, thank you very much.